In my research, I'm trying to find predictors for functional recovery in MS. So we're trying to find out why patients in MS differ in how well they can recover motor function. In the long run, this should help making rehabilitation more efficient and possibly even tailor it to the patient's individual needs. So we're trying to find factors that are beneficial for successful motor recovery. And today here at ACTRIMS I presented a study that I did at Cardiff with Dr. Valentina Tomasini, and we used an experimental approach to look at functional recovery and underlying brain plasticity in MS patients. So that means we didn't use a clinical intervention, but a highly controlled experimental setting where all patients had to do the same protocol and they had to do a motor sequence task. And I showed that advanced brain imaging measures, in this case it was fMRI, could help predicting the outcome of such a training. So in particular, I showed that adaptation of brain activation during such a task over a short time scale could predict the outcome of a longer term training in the same task. And that on top of factors like clinical and behavioral measures could explain variability in the rehabilitation potential for participants. So we're not looking at treatments per se yet. This is more of a background research project. So the assumption is that when participants rehabilitate and regain motor skills, part of that is due to the fact that the brain is plastic and rearranges its structure and its function. And by using an experimental approach, we're trying to indirectly measure how plastic the brains are in the patients, and to see what factors are beneficial or can predict plasticity and recovery. So we want to find out in which areas we could target potential therapeutic approaches to then make rehabilitation more efficient. No, I'm not saying it works better. It's one way of measuring plasticity. With fMRI, we can measure functional plasticity, so changes in brain activation. And today I just presented some results of that included fMRI, so short-term changes in activation. But there's obviously a lot of other MRI measures that we can use as well, and they're obviously important. No, so the reasons we found to be involved in the short-term adaptation, they're quite well known to be involved in motor learning and motor sequence learning, just from studies with healthy human controls. So that's not unexpected. We still need to better understand what processes are exactly happening. So MRI is a good measure because you can use it in living human beings. But it doesn't tell you exactly what's happening on a cellular level, for example, and it wouldn't tell you which medications or which pharmaceutical strategies would benefit these kind of adaptive changes. I collected quite a lot of data that I haven't analyzed, so the next steps would be, as you said, to look at other MRI measures. So structure of the brain, also microstructure, and then MS-specific markers such as where the lesions are, how big they are, things like that. So as a long-term goal of this particular study, I want to include all the information I've got to like a bigger statistical prognostic model to then see which of these are the most important factors to explain why patients differ in training improvements.